Thank you very much, and thanks for the invite. It's yeah, really great to have an opportunity to come here. And I'd also like to start by acknowledging the Ghana people, which is where Flinders University and SAMRI are based, which is where all this university was acknowledged, or sorry, where all of this work was conducted. So yeah, I'm Feargal. I'm an NHMRC investigator. I was at SAMRI from 2018 to 2024, and then I moved to Flinders last year to start my own team. And the work I'm going to tell you about today was primarily work that I did at SAMRI in Professor David Lynn's team, which I'll talk about David's team in a little bit. And so I'm primarily a bioinformatician. My background was originally microbiology. And then I was just saying this about kind of career trajectory. So I very much ended up in Australia by accident. It was never really planned. My partner is a clinician and working as a emergency doctor in Ireland wasn't very good. So we decided to move out to Australia. And I kind of fell into a lot of this work by accident, basically. But you kind of just have to take opportunities as they come. And I worked very hard and got very, very, very lucky getting involved in this work. Um, so for today's talk, giving the audience that's here, I decided to kind of split it into two. So I'm going to first talk about this study that was published in Nature earlier this year, where we basically found that antibiotic exposure can impair responses to vaccination via the gut microbiome. And then for the second part, I was going to talk more just generally about multiomics and kind of, I've been trying to put together the sort of pro tips for multiomics analyses because multiomics integration is basically what I've been doing for my entire academic career, actually. That was my PhD. It's been every postdoctoral position that I've been working in. And I think through that, I've made a huge number of mistakes, but I've kept learning from them, I hope. So I'll hopefully try to communicate some of that to you there today. So starting on the vaccine work. So first, just to acknowledge all of the co-authors on this paper. Like I said, this work was done primarily in David Lynn's lab, but a very close collaborator who also was central to this work, Dr. Helen Marshall at the Women and Children's Hospital. And particularly to acknowledge Michelle, Miriam, and Saoirse, who really just did an enormous amount of work on this as well. But it was a big team effort from everyone here. Um, so when I first started working in David's team, they initially hired me because they kind of needed a, multi-omics microbiome person, basically. David's lab does a lot of work on vaccine responses. And this was back in 2018. And I didn't really know very much about vaccines. So I kind of came into the lab and I learned that, oh, vaccine responses are very variable across people. I was like, huh, I was sort of under the opinion that vaccines simply worked, right? And of course, skip forward, COVID pandemic happened. Everyone gets a real crash course in vaccines and how variable the responses can be, how some of us may get, have kind of more reactogenic responses. Some of us won't. Some of us will get really robust immunity, others won't. But it's not just true for COVID vaccines. This is true for every vaccine, okay? So on the left-hand side here, there's an excerpt from the Australian National Immunization Programme up to 4 years of age. So infants will get a Hep B vaccine offered in the hospital right after birth. Then at 2 months, they'll get three other vaccines. Infanrix Hexa, which protects against diphtheria, tetanus, and pertussis. It's called Hexa because there's 6 antigens in there. There's multiple antigens for pertussis. There's then and also targets Hib B and polio as well. There's the Rotarix vaccine, which is a rotavirus vaccine. It's oral vaccine. And then there's Prevnar 13, PCV 13, which is against pneumococcal disease. So that's against strep pneumoniae, a bacterial pathogen. And then there'll be different boosters to these over time. Is basically the responses wane, and so they need to get multiple boosters in order to get these infants to a sufficiently high antibody response that they'll be protected. But the antibodies are really, really variable against this. So against Hib, Haemophilus influenzae type B, which is administered as part of Infanrix Hexa, you can see up to 40-fold variation between individuals who get that vaccine. That actually is also the same for the pneumococcal vaccine. So remember that those two are variable. We'll come back to that. Antibody responses also for the seasonal influenza vaccine vary up to 100-fold between individuals, okay? So this is really very, very variable responses. This review from Nigel Curtis covers that quite nicely. But despite this, the vaccines do work. They're an extremely important part of our public health strategy. But if we're able to reduce some of this variability and improve the responses, even just a little bit, it can actually have really substantial consequences on a population health level. And so for the past kind of decade or so, there's been a growing body of evidence that part of this variability in antibody responses is due to the gut microbiome. or gut microbiota. So the gut microbiota is a really key regulator of the immune system. And we know this particularly when we look at germ-free animals or for kind of other indications that we can do in particularly mice. So this paper in Cell Host Microbe was published in 2018, just before I joined David's team, and it came out of David's lab, where they basically exposed mice to antibiotics in early life and then vaccinated them with some of those vaccines that I showed you earlier from the National Immunization Program. And basically, the mice that got the antibiotics had worse responses to vaccine. Around about a year later, this paper was in Cell from Valley Pullendran, who's a lab based over in the US, where they actually randomized healthy adults. I'm assuming these were probably PhD students that they recruited at the university to antibiotics prior to an influenza vaccine. And so it was a small study that it was randomized control design, so very strong from that point of view. But they ended up doing very deep omics on these people to show basically the immune pathways that were involved in the what was driving that. And so that was really nice evidence because that was actually causal, right? It wasn't just a correlation, it was in humans, we can impair the responses by giving antibiotics. And then there's been quite a lot of correlation-based studies in this space as well. So this paper in Gut as well from a few years ago, there was quite a lot of these studies, particularly around SARS-CoV-2 vaccines for obvious reasons. You know, so many different medical centers were setting up studies to look at this. And so naturally, kind of different researchers would add on bits and kind of work together to look at some of this, particularly because collecting fecal samples is it's an easy biosample to collect compared to some at least. David and the team covered a lot of the evidence for this in this article in Nature Reviews Immunology from a couple of years ago, including outlining some of the potential mechanisms for how this might be taking place. I suspect, depending on the vaccine you're looking at, you will see kind of different potential ways that it's happening, and you may even start to see a combination of these in some cases as well. So this kind of leads me on to the main study I'm going to talk about. So the antibiotics and immune responses study, or the AIR study. So this was a prospective observational study that we started here at the Women and Children's Hospital in Adelaide, South Australia, back in, I forget when recruitment started. It was late 2017, I want to say. Where the idea was that we would recruit healthy, full-term, vaginally delivered infants who got some level of antibiotic exposure. The antibiotic exposure groups would be defined a priori. We were blinded throughout to everything. So infants would, by the clinical team at the hospital, be slot into one of these exposure groups depending on what they got. Excuse me. So, for example, one of the most common exposure types that we ended up getting was group B strep prophylaxis. So, mom comes in to give birth. The clinicians aren't sure of what her group B strep status is. So, she'll be given prophylactic antibiotics that baby might be exposed to because it's in that intrapartum window. One of the other ones we get a lot in this study was early onset sepsis exposure, or sorry, suspected early onset sepsis, right? Where clinicians are extremely nervous about early onset sepsis in neonates. So if there's even a potential chance or sign of something, they'll give 48 hours antibiotics like that. And actually, for a lot of these infants, they're completely healthy. That's culture negative. They never develop any other symptoms. They're totally fine. And so we were leveraging that. It's, I guess, described as antibiotic overexposure to kind of look at the effects of antibiotics in a healthy cohort. And so this comes down to our four groups that we ended up having. Our no ABX group, our infants who got direct exposure in the neonatal period, which is neonatal antibiotics. The intrapartum antibiotics is really kind of bystander exposure, where mom is getting the antibiotics in the sort of 48 hours around birth. And then what we have is postnatal maternal antibiotics, where mom is breastfeeding and takes antibiotics. So again, the infant can be exposed kind of subsequently. And so really what we were interested in doing was looking at these early time points, looking at how the microbiota develops in kind of the first 6 weeks of life, where we ended up sequencing over 200 samples, or sorry, is over 200 people and then 2 samples per individual. From week 1 and week 6. And then we also collected bloods actually from these people right out to month 7 and month 15 of life, okay? So what we were end up doing was kind of trying to relate the microbiome in these first weeks of life to the antibody responses 7 and 15 months later. So right around week 6, week 7 was when infants got those combination of vaccines that I said, Infanrix Hexa, Rotarix, and Prevnar 13. And it was around this time we also got blood for transcriptomics too. and also flow cytometry or single cell flow cytometry to look at that. And so, yeah, from our omics there, so we had over 400 metagenomic samples, 300 bulk total RNA-seq, almost 500 samples for antibody responses, and then 150 for flow cytometry too. So the kind of top level finding from this was basically that those infants that received antibiotics in the neonatal period, and this was 48 hours of antibiotic exposure for suspected early onset sepsis, almost universally across the whole cohort, that's what it was. Basically, these infants ended up having significantly impaired responses to multiple vaccines 7 months later. So these first two plots are from the Prevnar 13 vaccine that has 13 antigens in it for different serotypes of Streptococcus pneumoniae. And then this last one here is for Haemophilus influenzae type B. So these again were those ones with the 40-fold variation between individuals. And what we can see is the infants that had received the direct exposure, not just actually had significantly lower responses. In many cases, these infants are now below the threshold of protection. So they're no longer achieving that, what's called a seroprotective response from these vaccines. This is all of the antigens covered. So one thing, it's always a challenge with these big data sets is finding a nice way to visualize it. For visualizing the antibody responses, we ended up using this. So each column is a antigen from one of the vaccines, which we've colored by the vaccine at the bottom. And then we've represented it here as a fold change across our different antibiotic exposure types versus our NoABX group. And what you can see is that where it's white, there's no change relative to the NoABX controls. Red means it's higher antibody titers in the NoABX controls, which we observed one instance of, didn't quite reach statistical significance though. And then everywhere it's blue, it's basically lower. And when it's deeper blue, it's quite a bit lower. So you can see really across the board that was a very consistent signal that we detected in some cases for kind of any antibiotics, but really it was driven by that neonatal antibiotic group. In order to understand what was driving this, like I said, we got these blood samples for bulk total RNA-seq, where we did some very deep RNA sequencing on these samples. And basically what we found was that in the infants that were exposed to antibiotics, that we started to see this pro-inflammatory like signature pre-vaccination, actually, okay? So these infants had higher level of transcript associated with neutrophils, inflammatory signaling, TLRs, monocytes, all innate immune activity. And then comparatively lower expression of genes related to B cells, your adaptive immune response that's responsible for actually making antibodies, right? Interestingly, when we look at how the infants responded to vaccination, we didn't see any difference in the actual response. But because these infants started from a different baseline, they ended up actually kind of again at a different baseline, even though they still had a quite robust response to the vaccine, right? What you can see down here at the bottom right is from our flow cytometry data. So we developed these flow cytometry panels where we basically looked at different immune cells in blood. And this was one of my co-authors, Sirsha, who painstakingly freshly processed every blood sample as it came in from the hospital. And I think was a little disappointed to see that we found almost no differences in the single cell data. Which it was extremely important data to generate and extremely important to do it right. But it's always challenging when you get a negative result like that. But the data set itself was extremely useful for integrating with the blood RNA-seq because we can, of course, disentangle some of the bulk signatures using the single cell data, which was quite nice. And that's what's covered there. So then, like I said, we had this big metagenomic data set here where we ended up doing bacterial load alongside this. So we could kind of do both relative abundance and actual more of a quantitative type approach. I won't go into too much of the details from this, but basically at week 1 of life, we found all of these decreases and increases in different groups according to their antibiotic exposure. By week 6 of life, week 6 being the time they actually get their vaccines, almost all of these were gone, okay? And one of the only things that remained was a decrease in Bifidobacterium in the infants that were having the poor responses, these neo-ABX infants. And you can see some of the individual Bifidobacterium strains plotted up here. You can see this quite significant drop up to week 6. And actually, when we look at the counts of these Bifidobacterium, they're directly correlated and statistically significantly correlated with the antibody responses 7 months later. Okay, so it's not just lower. There seems to be, in some regards, a linear relationship from this. I'm not going to talk too much about this today, but then some of my co-authors and colleagues there did an enormous amount of preclinical work as well in germ-free mice and SPF mice to basically, again, demonstrate causally that you can actually remove the microbiota, put back in the microbiota, and this is able to adjust the responses accordingly from that. So just for this part of the talk, just to again acknowledge my co-authors and the funders from this. So that's the Lynn Lab there, and this was one of the laboratories during COVID. We couldn't leave the state, but we went off up north for this, which was quite nice. And this work has now led to a clinical trial where we're actually testing probiotics to improve vaccine responses in infants. That's funded by the Women and Children's Hospital Foundation here in South Australia. Um, so on that, I'm going to take the last, I think I've about 10 minutes or so, according to my counts, to give some tips and kind of multi-omics analyses. So like I said, this is basically what I've been doing my whole career. So my PhD was this, and all of my postdoctoral work has been this. I went for this talk, went to count up how many samples that was. So it's about 15 clinical studies I've done this in, around 8,000 samples from 2,000 people from cohort studies done in Australia, Europe, and North America. This covers transcriptomics, proteomics, lipidomics, everything. Included within this that functionally acts as an omics there is also stuff like dietary data, flow cytometry data, cytokine data, antibody responses, T-cell receptor sequencing, and so on. And then, of course, there's also a bunch of that work I've done across preclinical studies and a bunch of in vivo and in vitro systems. Although I actually think the multi-omics analysis in a preclinical system is it's obviously challenging, but it's much easier than the comparative is in the clinical data, just because humans are so bloody variable in kind of every regard, right? And like I was kind of saying earlier, I made so many mistakes throughout my career in this, but I always kind of try to keep learning and try to, build that up over time, which I think is good advice for anyone in any career. So my first real top piece of advice for multi-omics integration is basically this kind of meme from The Office, which I always tell people, I always reference this. Um, and it's a quote from Dwight where it's keep it simple, stupid. Great advice, hurts my feeling every time. So when you're dealing with these big multi-omics data sets, the number of features you have, it gets really, really huge. Start with straightforward, simple hypotheses, basic things, and build up from there. So simplest approach of all, you might want to cluster your patients based on a microbiome profile and then use those in your transcriptomics analyses for differential expression analysis. That's very, very, very simple, but it is on a fundamental level multi-omics integration, right? Although it doesn't always feel like it because you're just kind of taking simple group labels, right? Multi-omics data sets open a lot of questions. You don't need to answer all of them, okay? Your goal is to tell a clear story and specifically tell a clear story in a paper that depending on the journal you're going to, there will be a word limit, there will be a figure limit, there will be a table limit. Overloading it is going to make it too hard for the reader to understand. It makes it hard for the editor to understand. You also want your work to be easily citable. Let's face it, people are going to look to make a point and they're going to type something into Google and probably one of the top results they see is the citation to put into their paper, okay? So you want your findings to be easily understandable, and just keeping it simple, I think, can help with that. Your interesting side things aren't going to be lost. You can always return to the data later, okay? No one's going to be sitting on the SRA waiting to scoop it up. So keep it simple, focus on the hypothesis and the question in front of you. Don't try to do it all. Two, this was something I've had a lot of my colleagues get caught up with this. They always feel like the data needs to be consistently processed in the same way in order for it to be comparable. And that's not true. You want to address the technical challenges of each omics layer separately. Okay. So different data sets will have different requirements based on how they were processed, how noise needs to be removed, how you deal with batch effects. This is particularly true for sequencing-based omics, RNA-seq versus mass spec-based lipidomics or proteomics or whatever, right? Taking even similarly related omics, okay? RNA-seq and metagenomics are very, very similar in many, many ways. You even have RNA-seq tools get repurposed for metagenomics, but there's some really crucial differences. In human RNA-seq, you have a really fantastic reference genome. You don't have a really fantastic reference genome for your metagenomics, most of the time at least. Even in the human microbiome, which is a really, really well-studied system, there's going to be things missing, so you use different normalization approaches. Similarly, the level of sparsity, that is zeros that are detected in the microbiome data, is much, much higher than what you see in RNA-seq, where you have actual housekeeping genes that will be consistently expressed across samples and cell types, okay? So this changes how you actually analyze your data. To say nothing of the fact that RNA-seq, you can have alternative isoforms, which are, again, quite different to what you might see in microbiome data. So just take the technical challenges of your data separately and use the best gold standard approach for your data at the time that you're working on it. Next, narrow your feature space. Again, this kind of goes to what I started with. There's so many possible combinations of features. Even on the largest multi-omics data sets, you're not going to be powered for multiple testing correction for tens of millions or even hundreds of millions of tests. So my kind of advice is don't even try. Focus in on the questions you want to ask. So, or sorry, if you want to take it like an unsupervised approach, you can do that. So you can, for example, remove features that are very lowly detected or with very low levels of variance. Dimensional reduction techniques are extremely powerful in this space, and mixomics is really, really fantastic in that regard, particularly with the level of documentation that team puts in is really, really A1, so that's really great. Cluster of clusters analysis, not as widely used, but really cool approach that we used early on in the TCGA. You basically just create clusters of each omics layer, and then you operate on the clusters, which you can just do with simple Fisher's tests or hypergeometric tests, which is nice. You can also use supervised approaches. I don't see this as commonly either, but again, it can be quite powerful. So for example, if in your transcriptomics data, you're only interested in the adaptive immune expression genes, like if you were doing a vaccine study, for example, you can use something called gene set variation analysis that converts your gene X sample table into a gene set example table. So you can go from whatever 15,000 genes down to 10 or 15 gene set features with the values then becoming how highly or lowly they are expressed across samples, which can be quite powerful and a nice way to narrow down that feature space. Or you may simply only focus on genes that are differentially expressed across your condition, and that are a very simple way to narrow your feature space. Decide early on how to handle your missing data. Again, straightforward. Sometimes you just have to drop features. It's okay. If the data is mostly missing or if it's missing unevenly across groups, I would say simply exclude it. Imputation works in many cases, but I never want my findings to be entirely dependent on an imputation. So I'll kind of always do it with and without, which maybe then it's not even worth doing, I guess, if it's with and without, if that makes sense. The context of the missing data is very, very important. So what's missing in mass spec can mean something very different to what's missing in a questionnaire, where maybe is it below the threshold of detection, or is it because the younger participants are skipping a question or questionnaire? So the meaning, what does the missingness mean? Think about that before how you decide to deal with your data. And then 5, and this is by far the absolute most important piece of advice that I'm constantly telling my students and everyone else that I work with, which is even though we're data analysis people, we need to know the biology. Okay, so many of us love the coding, we love the analysis, we love the data visualization. We don't always spend as long reading the literature, which is often, I think, it's the most common weakness I see in kind of early career bioinformaticians, particularly. Knowing the biology, for me, I think it is not just an important part of the analysis, it is possibly the most important part of the analysis, right? Because it enables you to sanity check your data. So you don't need to be an expert in everything, but you need to know enough. So for example, if analyzing your single cell RNA-seq data, you don't need to know all the biology, but you need to know enough to make sure that your cell annotations make sense. Is the ratio of T cells to monocytes right for the sample that you're looking at, okay? Working with collaborators is a bit of a shortcut to this, but ultimately someone has to know enough of both to actually be able to interpret that, okay? I found like popular science stuff is a really, really, really great place to start. There's this really fantastic YouTube channel that does these really nice videos, particularly about the immune system. I find their stuff is great there. And they have a book as well called Immune. It's very, very great for kind of basics of immunology, which is particularly good, I found, for students that kind of come from other backgrounds. And now, they're analyzing a bunch of single cell RNA-seq. If you're going into the microbiome space, I Contain Multitudes by Ed Young, really, really nice resource. There's a couple of other books in that space as well. And The Emperor of All Maladies, really, really great for cancer. I absolutely loved that read. This is really important for publications. If you can demonstrate to the reviewer that your advanced multi-omics integration reflects known biology, it makes the novel biology more plausible, okay? So again, it's just really, really important for telling that story. In the context of our vaccine work, so we actually have a bunch of sub-data showing the waning of the immune responses over time. This is a known thing that happens with vaccines. And then also a known thing with vaccines is that the pre-vaccination antibody titers are inversely correlated with post-vaccination antibody titers. So by demonstrating that, it made our novel finding more plausible. Similarly, in our metagenomic data, we know for infants at this time point that the alpha diversity should be increasing over time, and we're able to demonstrate that, which again makes our novel findings more plausible. Kind of fitting into this as well, if you know you have a particular genotype or chromosome-specific association, look for it in your data. The presence of a Y chromosome or X chromosome inactivator exists can confirm that your samples will match their male and female labels for your metadata, really important. And then also just leveraging established biology. Molecular signatures database is really great. Again, in our vaccination study, are we seeing an adaptive immune response signature? Are the bacteria you're detecting consistent with the environment you're studying? I've lost count of the number of papers I've been asked to review for major journals where the bacteria they're reporting is different, is, an extremophile only ever detected in, Yellowstone National Park hot springs. I'm like, well, this isn't a human gut microbe. I can tell you, something's wrong. Something's gone wrong upstream in your analysis pipeline. Looking at housekeeping genes in transcriptomics data to help make sense of your data. Across omics data, across omics, there are you detecting biological associations that make sense between a transcription factor and its targets, for example, or between a transcript and a protein? Leveraging that sort of biological ground truth is going to be able to help inform your downstream omics analyses. So I'll wrap up there. Just in summary, keep it simple, address your technical challenges, narrow the feature space, know how to handle your missing data, and for the love of God, know the biology, please. And with that, I'll wrap up. Thanks.